Good morning, everybody. We're done with the ecological models. We're going to start talking about molecular dynamics, and today's topic is possibly the most important molecular model, dynamics model. Have you guys heard of enzymes? What is an enzyme? Abdon.
They speed up reactions.
It's a molecule that speeds up a reaction. That's right. Does any molecule that speeds up a reaction, is it called an enzyme?
It's called catalysts.
Could be a catalyst, okay. Okay, so catalysts are molecules that help speed up reactions. An enzyme, I guess in that case, would be a type of catalyst, okay? And what do catalysts have all in common? Can you use up the catalyst in the reaction?
No.
It doesn't get used up, okay? So it's a molecule that participates in a reaction, makes it possible, but does not get used up on the way, okay? So let's think about it. Now we're going to start talking about molecular models, not ecological models. So for the beginning, we can think about it in the same terms, though, as we talked about ecological models before. Let's think about molecules jumping around in space, in some fluid, okay? And they're bumping against each other, and every time they meet, every once in a while they meet, and then they react, okay? They bind, they do the wrong thing. So suppose that I'm going to give you the following example. This is section 7.1 or 7.2, something, 7.1. Okay, so example we're going to talk about today is food transport. And I'm not talking about UPS or Whole Foods. I'm talking about transport of food into a cell. Okay? So you have a cell. This is not drawn to scale. And there's a bunch of food that's outside the cell. Okay? And the food, ideally, should make it into the inside of the cell. Now, the cell has a membrane. The cell has a membrane that protects it from the outside world. So not everything can just flow right through the membrane. Otherwise, there would be no point to having a membrane. The membrane makes it so that everything that, well, a lot of the stuff that has to go in and out has to be transported. It doesn't just go by itself. So there are these receptors that bind to the food. And once they bind to the food, they magically, I mean, you can actually look into the mechanism for this, but they actually flip around the food, they bring it into the inside, and then let it go. Okay? We're not going to talk about the mechanism of how this thing lets it go inside. But let's just imagine that once the membrane, once the receptor is bound to the food, it can go either back outside or it can be inside. Okay? But it has to bind to the receptor before it can go in. All right? So now let's think about how we can write this reaction down. So I'm going to call S the external or extracellular food concentration. Okay? You can also talk about extracellular food numbers. You can also think about the units being the number of molecules. But in the case of cells, you know, it really depends what boundary we have. You know, if we have the plate go all the way up to here, you got a very different number from if you count the whole, you know, all the way up to here. So instead we talk about concentrations. Okay? It doesn't really matter how much food there is overall in the plate, it just matters what the concentration is. Okay, we're also going to have the receptor concentration. Concentration. But we're going to differentiate between two different types of receptors. There's the receptor bound to the food, and there's a receptor not bound to the food. Okay? Because it can be bound or unbound. This guy is bound, this guy is unbound. So this says E0 is unbound. Okay? And E1 is receptor bound to the food. And finally, we have we're going to call this P, is the intracellular food. Concentration. Okay. Once again, don't get too caught up on the concentration thing. You can think about it as a total number if you find it easier. And then the math is pretty much the same. It's really the same. Okay. So who wants to try to model this mathematically? Let's think about it a little bit like the molecules we found in like the models we found in ecology. So these molecules are bumping around, and every once in a while they bind together with the receptor. Okay, we're actually going to use a chemical notation. We're going to think about this at first, like very intuitively, and then we're going to start making more sense of it. I think in the next few lectures, we're going to be a lot more precise about what these reactions exactly transfer to mathematically. But first, we have a reaction. What is the first reaction you can think of? The first reaction, what happens first?
Food binds to a receptor.
Exactly, the food binds to the receptor. So how would you write that chemically? The food binds to the receptor and turns into what? Turns into E1, that's right. Okay? In a moment, we're going to write this down in terms of equations, in terms of ODEs. But for now, let's just think about the reaction. Now, we're going to assume that the food can bind and unbind without actually going in. We're going to let the food bind and then, just unbind. It doesn't matter. Just every once in a while, that might happen. So this reaction is reversible. It also goes the other way. And what other reaction can happen?
E1.
Okay, E1 turns into what? P
Plus S. Oh, but that's
plus what?
it couldn't be plus S because that's extracellular.
No, not S. Something else. Remember, plus is 0. That's right. Because remember that the enzyme does not get used up. So when everything is said and done, you end up with the same amount of enzyme you started with, right? So P plus is 0. Okay. All right. So we're going to give these things parameters. We're going to call this parameter K1, this parameter K minus 1, and this parameter K2. And these parameters are going to describe the speed at which each reaction takes place. We're going to see in a moment how you use those parameters. Now we're going to transfer this into ODE form. Maybe I'm going to write it over there. So we have S of t, we have E0 of t, and so on and so forth. So each of these variables is a variable over time. We're being a little bit, chemists might not like what we're doing because we're calling S, the function S, we're giving it the same name as the molecule S. Sometimes chemists would write something like concentration of S. But we don't, those brackets, I don't really like them, and neither does Leah, because you're going to start seeing brackets all over the place. The more math you do, the more brackets you're going to start seeing, so you just get rid of the brackets and that's it. Okay, so no brackets. You're probably not even going to get confused about this unless you're chemists. So we call S the molecule, and we'll call S the concentration. Okay, no problem. So what is now ds dt? Well, let's think about it. What reactions is S involved in? How many reactions is S involved in? Two reactions, right? This reaction and this reaction. This reaction destroys S, right? I mean, it binds it, so it's not there anymore. So the first reaction has a minus sign on it because it's reducing S. Okay? Now, from what you guys know from rabbits and wolves interacting, how would you write this reaction down? The rate at which this reaction happens is what?
Depends on the concentration of S.
Exactly. The more S there is, the more the reaction takes place, right? The more E there is, E0, the more the reaction takes place too. So then what's the easiest thing to do?
K1 times S.
Exactly. Minus S times E0. Okay? That way, the higher the value of S, the faster this reaction takes place. And the higher the value of E0, the faster it takes place. And because we still want to find out how fast the reaction is happening, there's this parameter, right? Remember we said we always put parameters in front here because otherwise the units would not even work, right? So here we write K1. So that means if K1 is very high, that means that these molecules really, really want to get together. And if K1 is little, then these molecules don't really get along so well. They don't bind so much. So K1 gives you like the nature of the binding. Is it a very good binding or is it not such a good binding? What about the other reaction? Does anybody want to try? For example, Nelson? Is it plus or minus?
Plus.
Why plus? Well, this reaction is producing S, right? When the reaction happens, S gets produced, so there's more S. So.
No, it's plus.
No, you see when the reaction happens, S grows, so it contributes to the growth of S. So S plus S time is, the rate of change is positive because of that reaction, right? So plus. Plus what?
E1.
Okay. Multiplied by something?
K subscript -1.
That's right. Okay. Now, this reaction right here, as a matter of fact, does not depend on S. This reaction is the reaction where this receptor bound to the food spontaneously decides to unbind. It doesn't really matter how much food there is outside for that reaction to happen. It only depends on how much bound receptor there is, right? Okay, now let's talk about E0. Let's do the same kind of thing for E0. Now E0 is involved actually in the same kind of reactions as E1, as S. So this would be minus K1 SE0, okay, plus, what do you think? Plus K minus 1 E1, okay? But there's actually another reaction where E0 shows up. The third reaction, right? Okay, so that means that this reaction with rate what? Daniel.
K2.
K2 times? K2 times? Times E1. It's the same as we were saying before. The rate of this reaction doesn't actually have to do with how much product there already is. This reaction corresponds to the spontaneous unbinding of the food into the cell, right? And it only depends on how much bound receptor there is. So K2 E1. All right, let me go a little faster on this one. E1 prime, that's the bound receptor. So what reactions are involved in this molecule? What reactions involve this molecule? All of them, right? Let's talk about the first reaction. The first reaction makes E1 go up or down? Up. Okay, so now it's plus K1 SE0. The second reaction, does E1 go up or down? Down, that's right. So it's minus K minus 1 E1. And the third reaction, does E1 go up or down? Down. So it's minus K2 E1. Okay, and finally the product. Where is the molecule P involved in which reactions? Only one, right here. Okay. The only way that food inside goes up is when this reaction happens, and the rate for that reaction we already saw is K2A1. K2A1. question. Wilson.
Is the E0 and E1 supposed to denote when it has one and it doesn't have one, or?
Correct. That's the definition, right? E0 is the receptor concentration, but only the unbound receptor, and E1 is the concentration of the receptor that is bound to food. Okay. All right. Now we're going to spend a few lectures talking about chemical reactions. So at some point we're going to learn a systematic way to go from here to there. But for now, let's just think about the feeling of it. Just, write it down and think about the intuition behind it. So there's a concept regarding that kind of system that is quite important. It's called mass conservation. Okay? As it turns out, there's a conservation process. Here, molecules are not really being destroyed or produced. They're just being transferred and transported. Okay? So let's try and write down what we mean by mass conservation in this case. What is there's two types of receptors. There's receptor bound to the food, and there's the receptor unbound. So what will be the total receptor concentration? Counting everything. What is that? Okay, so total receptor concentration is E tot. We're going to call this E tot is equal to E0 plus E1. Now let's think about this intuitively. You got these molecules bound on the cell. Food is binding and unbinding. Every once in a while it gets transported in. Is the receptor getting destroyed? It's not getting destroyed. It's staying the same. Okay? So what would you expect E tot would be? Is it changing over time? It's not changing over time. It's constant. Okay? So in fact, E tot prime is equal to E0 prime plus E1 prime. And look, minus, plus, plus, minus, plus, minus. It all cancels out, right? Is equal to 0. So you can think about E tot as a constant. So whatever you start with for E tot, that's the value that it stays with. It's not going to change. It's almost like E tot is a parameter of the model. You say, okay, you write all those equations down. What E tot do you want? Or what do you think? What do you want the initial concentration of receptor to be? And you set it, and it doesn't change. All right. What else doesn't change? Can anybody tell me another mass conservation property in here? Total food. Excellent. Now, this one's a little bit tricky. Let's write this as S tot. What is the total food? Michael?
It's the extracellular food concentration plus the intracellular.
Ah, nice. E1 has food too, right? Yeah. Turns out when you add these things up, right, they give you 0. If you just add S and P, it doesn't give you 0. Okay? I'm not going to write it down, but, well, I mean, we could write it down, but it's going to take a while. So SP and E and E1. This guy here, and this guy here. So this one cancels out with this one over here. Okay? And these two cancel out with these two. Okay? So the whole thing adds up to 0. All right. Great. Awesome. And that's going to be very useful. Why is it useful to know that the sum is a constant? Well, what do you think? If we know that these two variables add up to a constant, what can we do? Exactly. We can solve one in terms of the other. That's a very good idea, Wilson. Thanks. So trick by Wilson. Since E tot is equal to E0 plus E1, we can write E0 equal to E tot minus E1. And this sounds, it sounds silly until you realize what this means. We have a four-dimensional model, right? But suddenly we can write every single appearance of E0, we can write in terms of E1 in a parameter. That means we really got rid of E0. We're down to three dimensions instead of four.
Don't we add an E total, so it's added another?
No, because remember it's a parameter.
Oh,
That's the good thing about it. If it was another variable, you're absolutely right. It would just make things more complicated. But remember, E tot is constant. It's a parameter. So it becomes a three-dimensional model with the parameter E tot. Okay? It's a useful trick. Whenever you have conservation, mass conservation, you can do this kind of thing and get rid of variables in the system. Okay? And eliminate E0 altogether. Okay. Yes, Wilson.
I don't really get because I feel like, isn't that the E total statement saying that there's no E total equals 0?
Not E total, E total prime.
Oh, okay. So it's not changing. Okay.
Yes, the rate of change of E tot is 0. So E tot is really a constant over time.
And what about S total? That's not a derivative.
Oh, you're right. You're right. Thanks, Wilson. I made that mistake myself. S tot prime is equal to S prime plus P prime plus E1 prime is equal to 0. Thanks for pointing that out. Yep. Okay. All right, good. So now we're down to a three-dimensional model because you know where we're heading, right? We want to analyze that thing using the tools we learned, right? Can we simplify it even more down to two variables? What do you think?
Dimensionalization.
No, it's easier than that. It's actually easier than non-dimensionalization. We essentially can get rid of this one, E0. Okay. Now, notice that P. Yes, Krista.
We can get rid of E1.
How?
P prime divided by K2. Just by the loss equation.
Yes. Wait, you mean you can get rid of this one?
Well,
P-
we can plug in, so get rid of E1 by plugging in P prime over K2.
Yeah, I almost want to do it the other way around because you see, if we write here a P prime showing up on the right side, it's not going to be of the form time derivative equal to state, function of the state. There's going to be derivatives on the right. That's not the form we're used to having, right? So it would be easier to do. You're absolutely right in the idea. This formula is going to be very useful, right? But what this means is that if we know the dynamics of these two alone, and we know what E1 looks over time, then we can figure out what P prime looks, what P looks over time by plugging it into this equation. So the point here is that P doesn't show up in here and P doesn't show up in there. So the trick to simplify this to a two-dimensional system is very simple. Basically, ignore P. Right? I mean, look, the S and E1 depend on each other. They're coupled, right? But P does not show up anywhere in here. P is a downstream variable, right? So if you can find out what these things look like over time, then you can easily find out what P looks like over time. So we can also get rid of P by ignoring it. Okay? Since it doesn't affect the dynamics of E1 and S. Okay, so now we're down from 4 variables to just 2 variables, which is great, because 2 is like our magic number, right? We have a lot of tools to analyze two-dimensional systems. We can do null lines, we can do all these Jacobian stability things with the determinant and the traces. That is easier than the three-dimensional case. Okay, so it's great. Let me write down that system just for here. Let me write down over there. Yeah. So the 2D system is now the following. S prime is minus K1 times S times E0. But what was E0 again? That's right. Minus K minus 1 E1. And E1 prime is what? K1S times what? Okay. Minus. You know what? We can write this down as like this. How's that? All right. Wonderful. Yes.
I had a question about, is there a limit to how much a cell can absorb?
Good question. Is there a limit to how much the cell can absorb?
Then P would then affect S if if it was too much, then they would slow down their absorption.
Can you just assume it's
That's-
reduced at the rate that it comes in or something?
Well, but he's got a point. If P is if the cell starts to fill up with food and it doesn't let the food in anymore, then that's going to violate the model because the model doesn't incorporate that. So Krista is saying Wilson is right, and we can just assume that P kind of feeds back on the remainder of the system to prevent it from coming in, right? But that's going to make the model more complicated. Okay, we don't want to make the model more complicated at this point. So Wilson has pointed out to another very real problem here, or another possible issue, which is that if there's a ton of food outside, eventually it's all going to start filling up the cell, and the cell's going to blow up. Okay, so for the moment, let's think about a small enough amount of food that it's not going to blow up the cell. Okay, but that's a very good point, Wilson. And long term, that's going to be a problem. But let's look at that model from the point of view the food is just coming in. Eventually, all the food is going to make it in. For example, it's not very realistic, but that's For example, it could be a different type of molecule. It could be not food, but something smaller quantity that doesn't have that problem. Yeah, that's a good point, Wilson. I don't think that Leah says that in her book. I'm not sure. Yeah. Yes.
Isn't that an issue every time we see like exponential growth?
Good point. Whenever you have exponential growth, you are assuming that there's infinite space, which is not true, right? But you make that assumption just because it makes you a simpler model. Okay, great. Okay, so now we still have 20 minutes. So we are going to analyze this system, but not right now. In fact, not today. We're going to leave it at that. It's a 2D system. Next time we're going to analyze it. But first I'm going to tell you the significance of this. It's not just about food. Okay? It's much more than just food. And that's why we call this an enzyme model, because this is a model of any enzyme. For almost any enzyme can be modeled this way. Yes?
Sorry, just one last thing about that last part.
Yes.
Wouldn't that be where the carrying capacity comes into play? Or that kind of thing?
The carrying capacity for the, like how much food the cell can carry?
Right.
Yeah, for example. Yeah. So maybe there could be some kind of quote-unquote logistic model where the cell can only take so much food, right? Yeah, that'd be great, yeah. And I honestly, I haven't thought about it. Maybe you can come up with one. I don't know. It's Friday. All right. Okay, so this model is actually very general. Very general model of enzyme dynamics. Okay, so what is an enzyme? So definition, an enzyme is a protein that catalyzes a reaction, and then as part of the definition of a catalysis, it does not get used up in the process. Without itself being used up. Okay, so there's a lot of examples of enzymes in the body. There's enzymes all over the place. For example, say that Krista is finishing her coffee. Okay, so the sugar in the coffee makes it inside her and makes it. Let's just say makes it into the bloodstream and into the cells. And at that point, each molecule of sugar starts getting tagged by one enzyme after another. You first start attaching molecules called phosphorylations, and then you start chewing parts out, and eventually you're chewing it into smaller and smaller pieces until it comes to the point that it turns into smaller and smaller amounts of energy from different types of molecules. This whole process of grabbing a molecule and tagging it and chewing it up is made by enzymes, one enzyme after another. Okay? That's the process of basically digesting food. All right? Many other examples of enzymes, okay, they show up all over the place. But they all have a similar behavior, like this idea that they catalyze the reaction without being used up. So let's think about the same reaction as before. S is the substrate. I.e., the molecule that the enzyme acts on. Okay, so they kind of come in pairs, sort of like eigenvectors and eigenvalues. You have the enzyme and the substrate. Okay, enzyme could not be an enzyme if there's not a substrate. And a substrate is something that an enzyme acts upon. Okay? So E0 is the unbound enzyme. E1 is what we could call the enzyme substrate complex. The enzyme comes and binds to the complex and then forms this complex. So enzyme substrate complex. And P is what we call the product. So after the enzyme is done with the substrate, it turns it into a product. And the reaction is exactly the same as we had before. So S plus E0 goes into E1. Usually this is called E and C, the enzyme and the complex, but it doesn't matter. Let's just keep this notation for today. K1, K minus 1, and K2. This is product plus E0. E0. What are we going to do with this model? Why is it so important? Well, it's important because it describes the way that enzymes act on substrates. But here's the question that Michaelis and Menten were interested in. So if the job of the let me ask a question in terms of the food and the receptor, you know, the example. If the job of the receptor is to transport food, what is the overall transport rate predicted by the model? Okay, so basically the question is the following. You have substrate. The substrate is turning into food. I'm sorry, into product. Or let me put it this way. You have extracellular food. It's turning into intracellular food. And ultimately what you're interested in is what is the rate at which this thing is happening. Okay? And we're not going to do it today because we don't have time, but I'm going to tell you the answer to this. And the answer is the answer is the following. The rate of transport is roughly equal to a parameter. Okay, fine, let's call it Vmax multiplied by S divided by K M plus S. Okay, Vmax and Km are two parameters. So they're just two names. You know, you call them A and B, whatever you want to call them. This is the rough rate of transport at any given time. Okay? What does this mean? Okay. What happens when S is 0? What is the rate of transport? 0 over 0 is 0, right? Okay. So as S increases, the rate of transport we can call this the rate of flow into the cell increases. Okay, does that make sense? The more food there is outside, the faster the food is flowing into the cell, right? Okay, but what happens when S is very large? Does the rate of flow go up to infinity? Exactly. Rate of flow approaches Vmax. So let me draw this as a graph. This is the flow rate, meaning the transport rate from the outside to the inside of the cell. And this is, here is Vmax. Okay, and the flow rate starts at 0 for S equals 0, and it starts going up. So as S increases, the flow rate starts going up, but it does not go beyond a certain point. All right, now Km is a parameter that tells you essentially how fast it goes up. I don't know if we talked about this the last time, but what is the rate of flow when S is equal to Km, according to this formula? 1/2 Vmax, right? Okay, so that means when S is equal to Km, this is roughly Vmax over 2. This is Vmax over 2. Okay, so Km tells you how high S has to be for things to start saturating. All right, so there you go. This is the formula that allows you to understand the rate of transport as a function of the substrate. Again, we're not going to prove it today, but that is the idea. Okay. Do you guys have any questions? No questions. Okay. Does this make sense? You know that, this overall flow rate? Yeah. All right. Questions about enzymes? No. Yes, Wilson.
Well, look at the little notation about the why is that K negative? Is there a reason why it's not just K1, K2, K3?
Good question. This is considered to be basically the same reaction as K1, except the reverse version. So we call this K1 and K minus 1 so that it's clear that it's forward and reverse reaction of the same thing.
Okay, but the rate isn't the same.
It's not the same.
Okay.
It's not the same. That's also a very good point. If you might think that if you make this twice as big as this, then it's all the same in the end. I'm sorry, if you make this twice as big and you make this twice as big, that is overall going to be the same, but no, it's not. They're actually independent parameters. They all do different things. Yeah? Any other questions? Okay, great. So enjoy the weekend. See you.